and she's going to be telling us about microbial bioburden of bathroom door handles. Ooh, watch out for this one, particularly at Cal State College.
Yeah. Good afternoon, everyone. My name is Anna Martin, and I want to thank you so much for coming to my presentation today. It is on the microbial bioburden of bathroom door handles at Cal Poly Pomona. So first, I want to ask, has anything like this happened to you guys?
What do you see?
Oh, I'm trying to get the video to work.
Oh.
Sorry. 1 second. Don't mind me. Should be working. Ah, there we go. Oh, sorry. Don't mind me. So, that's happened to me, though I didn't quite make that face before. So today I'm going to give you a little background on my project, go over the materials and methods that I used, and reveal the results of my project. Now you may say to yourself, I wash my hands all the time, but is it really that important? Well, the Global Public-Private Partnership for Hand Washing reports that 3.5 million children die each year from diarrhea and respiratory infections. And 1 million deaths could be avoided if their parents and the ones who take care of them and themselves wash their hands. Now, it's not just a problem in undeveloped countries. 63% of female college students wash their hands after using the restroom, but only 2% wash their hands for more than 10 seconds. So, the goals for this project was to determine the microbial bioburden on door handles leading out of the bathroom, including if there was any coliforms present. According to the Michigan Department of Environmental Quality, coliforms are a class of bacteria that live in the intestine of warm-blooded animals and are commonly used as an indicator of fecal contamination. Also, we wanted to determine if there was any pathogenic E. coli that was on the door handles. So, my hypothesis, as I was going into this, was I thought that there was going to be a lot. Bathroom door handles, Cal Poly Pomona, I thought were going to be covered in a high count of coliform bacteria, including that there would be some diarrheagenic clones. So, building sample, I went across campus. Every Wednesday at around 11 a.m., starting February 15th of this year and continuing every week until May 10th, I swabbed the door handles of both the women's and men's bathrooms of the following buildings, including the 24-hour computer lab, the Los Olivos Dining Commons, and the Collins College, which is the Collins College of Hospitality Management. They also run our the Kellogg West and the restaurant at Kellogg Ranch. So it's important for them to be sampled. So now that I've given you some background on my project, I'd like to describe the materials and methods that I used. So the materials that I needed to collect were sterilized test tubes with buffered Peptone water as a carrying media, some sterile swabs, ice packs to keep it all nice and cool, and an inconspicuous box to transport tubes in. So for performing PCR, which I'll describe what it is in my methods, you need extracted DNA, some primers, which I use LAC-C and UIDA, and GoTac, which is a Tac polymerase. So next I'd like to go over the methods that I used, which included swabbing the door handles, differential plating, gram staining, and PCR and gel electrophoresis. So first, swabbing the door handles. I aseptically opened my sterile cotton tip applicators, dipped them into the sterilized Peptone water, and started to swab the door handles. I had to be very careful to make sure I got all of the door handle. After carefully swabbing the door handle, I removed the lid covering the test tube of buffered Peptone water and just put the swab in and bent it off to make sure that I didn't contaminate it at all with my hands. So now you've got a lovely completed sample. After collecting, I did incubate my samples for 24 hours at 37 degrees Celsius and then perform my next method. So my next method was differential plating, which is McConkey lactose agar. What it is it produces a color change in your growth if they're lactose fermenting. So we're looking for bacteria that live in your colon, like coliforms, that are lactose fermenting. So the next method I did was gram staining, which is a staining technique. It differentiates bacteria based on their cell walls, and E. coli is gram negative. So it'll come up as the red ones up there. So next I did PCR. So what is PCR? PCR is polymerase chain reaction. It is a method of amplifying DNA. What's amplifying? Basically, you make a lot of DNA from a little bit, and you focus on a particular gene. So how's it done? I combine Tac polymerase, which is a DNA polymerase that functions at high temperatures, nucleotides, which is the bases in DNA. I use my sample DNA as a template, and primers. So then I place it into a thermocycler, and it goes through the 3 steps of PCR, which is denaturation, annealing, and extension. So the primers that I used were LAC-C, which detects lactose fermenting, which would be confirming my coliforms. I did UIDA, which is for E. coli, and then I did a multiplex, which each one of those will detect or can be helped in partnership to detect a pathogenic E. coli. So, as far as gel electrophoresis, it is a method for analyzing DNA. You separate DNA based on fragment length because DNA is a charged molecule. DNA can then, the gel can then be stained, and then you can see these nice bands that should pop up. So, results. So, 84 samples grew on the McConkey lactose, so out of a possible 408, so it's pretty good. But 17 samples were confirmed coliforms, which were 2 from Building 6 in our women's, which used to be our old business building, 1 from the CLA, which is our classroom administration building, in the women's bathroom, and Building 3 women's bathroom, 13 women's bathroom in our commons, which is the dining commons, 9, which is our engineering building, both the men and women's bathroom, our campus marketplace, which is another place where we have like food on campus. It was in the men's bathroom. It was in our library's men's bathroom, and it was at the Collins College in both the men and women's bathrooms. It also was in our biotech building in our women's bathroom. So 1 sample was a confirmed E. coli. But it wasn't pathogenic, so that's good. But it was from our building which is our biotechnology building in our women's bathroom. So, so what does it all mean? Well, since there's no pathogenic E. coli present, but we did find E. coli and other coliforms found, so that means there's hygiene lacking somewhere on campus, including at the Collins College. So since hygiene is lacking and people are still not washing their hands, I'm going to give you some instructions on how to wash your hands. According to the CDC, you need to wet your clean running water, warm or cold, and apply soap. You want to rub your hands together to make a lather and scrub them well, and make sure to scrub the back of your hands, including underneath your nails. Continue rubbing for at least 20 seconds. Then rinse your hands well under water, and then dry it with a paper towel or air drying. So now your hands are clean. I'm going to show you how to open the door without getting contaminated. So you can use a paper towel. You can use your shirt or your sleeve. So what if you don't have a paper towel because they didn't sell one of these? Well, if you're lucky enough, it'll be one of the push open, so you can use your elbow, or you can push it open with your head. So I know there's limitations on my study, so I propose these areas to do further research, which includes swabbing additional times during the week, and also swabbing for a longer period of time to get more samples, testing paper towel dispensers, as well as to just make sure that those aren't causing your hands to get even more dirty, developing a marketing campaign to encourage hand washing across campus, and survey students on their hand washing habits and what would encourage them to wash their hands. So while only one bathroom was found to have E. coli in it, and it ended up being not pathogenic, it doesn't mean that there's not a health risk at Cal Poly Pomona. So students really need to be informed of the risks of not washing their hands, as well as proper methods to prevent contamination. Thank you. I'd also really like to thank my friends, Christian Sanchez, who took all the photos for me, as well as was the coughing person in the video, and she actually does wash her hands. Katie Steffen, who was the director of the video, and took the video. Brittany Villagrand and Melissa Ziegel also helped me out with collecting on several days. Most of the time it was just me by myself. And Dr. Shelton Rinda, who without him, this project would never have happened. So, thank you. Does anyone have any questions?
Do you know if the janitors actually clean the door counters from one day to the next?
Not that I'm aware. We actually were in there as the janitor was cleaning it when we were filming our video, and she did it. Yes?
So when you swap the door handle, you swap the outside or the inside?
Leaving, so like as you're leaving the bathroom, so the ones that people would grab on.
You swap the men's door handle inside as well?
Yep. I had to be really careful. Some bathrooms were easier than others.
So did you consider looking for things like snap instead, which are also very dangerous?
Yes. Yes, but Dr. Miranda's specialty is pathogenic E. coli, so we were kind of focusing on that, but that would definitely be a really good thing to look for, because I'm pretty sure I found a lot of staff too.
Another question?
You mentioned both men's and women's bathrooms. Were they equally?
Actually, yes. I was surprised because most of them walking in, the men's bathrooms would be like actually look dirtier, but as far as the door handles, there was no significant